Good morning, everyone. Welcome today to Scott Ayton. He's the head of the Translational Neurodegeneration team at the Florey Institute. Good morning, Scott.
Good morning, Jodette.
This is another of the interviews in our series with Florey researchers, and we always start off with a really nice question of tell us a bit about yourself. So, Scott, over to you.
Sure thing, but maybe before I begin, I just want to thank you for organising this series, Jodette. It's wonderful to reach out to Parkinson's community. The Florey exists for patients and their families and living with this disease, and so I just want to thank you for allowing us this opportunity to reach this community, and I hope post-COVID we can meet more in person as well. So a little bit about myself. I'm a researcher at the Florey, and I work on neurodegenerative diseases, and these are diseases similar to Parkinson's disease or encompass Parkinson's disease, where brain cells unfortunately deteriorate, and this leads to the symptoms. Other examples like Alzheimer's disease. And so we do research on laboratory-based research, so research on animal models or cellular models of disease, and also clinical research where we use imaging or we investigate things within the patient to see if we can understand what's going on with the disease and also how we can treat the disease.
Okay, that's very interesting. When did you first become interested in Parkinson's research?
I recall when I was doing my undergraduate training, I was doing more broad medical research, and I was thinking that all of the major discoveries in medical research had, at least recently, had been in cancer, and I thought that, okay, well, we've really made these major breakthroughs in cancer, but when I looked at the neurodegenerative diseases, including Parkinson's disease, I really found that we hadn't made any breakthroughs. And so I thought the next series of breakthroughs are going to have to be in this space of neurodegeneration. Unfortunately, I was completely wrong, and we've had a series of major breakthroughs with cancer, and we have not yet had those same major breakthroughs for Parkinson's disease and Alzheimer's disease. And you know that relates to a number of things, including funding and also the challenges in working within the brain. But I do believe that we can find breakthroughs, can find effective treatments, and can make those similar groundbreaking discoveries that have happened in cancer. So that sparked my interest back when I was an undergraduate. I found that Parkinson's disease was a really interesting disease. What was going on? The changes that occurred within the brain to affect the motor problems that occur within the disease, and also at a cellular level, the chemical changes that were happening within the brain. I think this was a very interesting set of circumstances, and one that I thought that we could intervene with to see if we could slow the progression of the disease. So I sparked my interest back then. I did a PhD in Parkinson's disease, and I've continued to work on that disease since.
No, that's great. Great insights into where the money's going and where the actual solutions have started to be found, particularly in cancer. And we're hoping to get some more money into Parkinson's research. So, could you tell us a little bit about the research you've been doing?
Sure. So I'm head of the Translational Neurodegeneration Laboratory, and that means we focus on translational research, and that encompasses two directions. One is we take translations from lab to clinic, and that means that we work on disease mechanisms, looking at the chemistry of the brain in the laboratory using animal models or cellular models, and we use drug discovery at that point, and we try and translate that finding into people. And conversely, we take findings that we can, by looking at the patient and looking at chemical changes that occur within patients using various techniques, we then try and understand why those changes occurred by looking at similar pathways and that biology within the lab. And so we have these two pathways of translation. My first, I guess, focus of my research was looking at a pathway or a chemical change in the brain of Parkinson's disease patients involving iron. Now, iron is very important within the brain, but sometimes it can be damaging. And so I was looking at a drug that could potentially affect or control the damaging aspects of iron while still maintaining its benefits. And so I did some some laboratory-based research and found that this drug could be quite beneficial. Yep. And then fortunately that drug then went to a early clinical trial where it did showed some promise. And then it's now undergoing a larger clinical trial, and we're expecting to see the results of that trial to come out in early next year. So that's an example of where we took some understandings in the laboratory and tested it out, showed the proof of principle, but the real demonstration has to be in patients. And so we've done some early clinical trials, and those later clinical trials we expect to be released soon. So there's a lab to clinic translational pathway.
Right. So assuming that the results of this latter clinical trial are positive in the sense of they show that the drug is effective, what are the next steps that we would normally undergo in the clinical trial process?
Sure. I want to be clear that I'm not personally involved in this project. This is the work that I did, the foundational work, and then so other people have taken it up from the clinical role. So it really depends on what the outcome is. If it's a clear benefit, then it's a different story that if it's a marginal benefit or no clear result, in which case we need to go back to the lab. We maybe need to change what the drug is, make the drug a little bit more effective, and go through that similar pathway as well. So there's a lot of unknowns at that point. I suspect we may need an even larger study. This is quite a large study, but there may need to be even further research to show that this is effective. It really depends on what the result is, and the drug company that is working on that, how they make those decisions. But ultimately, if it does show promise, it will encourage further research to develop drugs with even better efficacy or better performance. So we've got basically a proof of principle drug, and we probably can do better than that. But to get the investment to improve upon that, we really need to show that in principle, the first drug works, and then we can hopefully find a better one.
Oh, that sounds great. Sounds very promising. And other research you're working on?
Sure. So that was an example of lab to clinic, and we also do clinic to lab, which is often not as often as appreciated as an important pathway of science development. And so one of the ways that we can do that is that we can, for example, we can measure factors in the fluid that surrounds the brain of people with Parkinson's disease, and we can compare that to those same factors that in the fluid that surrounds the brain in people in ordinary people without any neurological disease. And in doing so, we can try and identify. We do this for two reasons. First, can we find a factor that we can use as what we call a biomarker, and this is something that we can use to predict whether someone's going to get Parkinson's disease or not. And there's another sort of complexity there. There are different types of Parkinson's disease. There is normal Parkinson's disease and variants of Parkinson's disease, which we call Parkinsonisms. And so we want to be able to differentiate that at an early stage, who's going to have a variant of Parkinson's disease or who's going to have what we would call like a normal Parkinson's disease diagnosis. And so we want to try and use these factors to identify that at an earlier stage. And also, we also want to know whether, by looking at these factors, we can understand the chemistry of what's going wrong in the brain of people with Parkinson's disease. And so we can take those findings back into the lab and then further develop on our theories and hopefully identify drug targets. So for an example, a recent work where we've looked at around about 600 or so people, the fluid that surrounds their brain, of people with a mixture of different Parkinson's disease, and compared those to controls. And what we found is that certain pathways related to inflammation are differently affected in people with Parkinson's disease and what we call atypical Parkinson's disease. And so we've demonstrated that. This is our work ongoing, and now we're going back to the lab to understand, well, why are these changes occurring? And by understanding those changes, can we design drugs that intervene in those pathways to see if we can slow the progression of these diseases?
No, that's fantastic. Fantastic insights to be able to find that path of inflammation. Okay, so what are the next steps for Dr. Ayton?
Well, research is always ongoing. We always want to discover more and improve upon what we have. I guess for us, as I've mentioned, a big unknown is the results of this clinical trial that happened early next year, hopefully, COVID virus pending. And if that works out, then I think that we go back to the lab and we work on even better drugs to see if we get some improvement, we can get even better improvement. And also we want to understand those inflammation pathways better that occur in people with Parkinson's disease. Inflammation is a very broad term, and there are lots of different types of inflammation. And so we want to identify particular pathways or particular types of inflammation that might be affecting the disease. And when we understand that, then we can hopefully identify those drugs that might be able to intervene in those pathways to see if we can dampen down that inflammation and disease, hopefully improve patients' lives.
Well, that sounds fantastic. Okay, thank you so much for your time today. It's really, really interesting research, and we wish you all the best. And I do look forward to meeting in person post our little lockdown period.
That would be wonderful.
Yes. Okay, look, thank you so much again for your time, and thank you to the Florey for all of the efforts. It's been a really fantastic series. Thank you.